Protein Dynamic Solutions, continues its efforts to provide the advantages of our system to the biopharma industry through a series of videos demonstrating the capabilities of our ProteinMentor platform technology. The gold standard for protein ID assessment is amino acid sequencing. Today's complex therapeutic protein designs require additional important factors that must be included in the evaluation, such as relationships between the amino acid sequence with weak and covalent interactions that govern secondary and higher-order structures. This relationship is critically important for multispecific and antibody-drug conjugates. As part of product-related impurities, one must also evaluate post-translational modifications, such as the extent of glycosylation, as well as degradation processes like deamidation or oxidation, including the tethered end and payload, which are often designed for antibody-drug conjugates. Also important is the formulation condition to ensure quality, safety, and efficacy. Our rapid screening platform method can evaluate all the above within a single experiment against a reference in just 10 minutes. The advantage of the flexibility of the slide cell array sample setup for developability assessment is the ability to define differences amongst an array of up to 21 lead clinical candidates to de-risk preclinical candidate selection. We can also evaluate the role of formulation conditions and excipients on the stability of the preclinical candidate. A first scenario involves formulation optimization, where various formulation parameters are evaluated to ensure physicochemical stability under varying formulation conditions based on their drug delivery. This is especially true for subcutaneous delivery, for which high concentration and high dosing conditions are required, and the concern for self-association or aggregation risk at high concentration needs to be addressed. The varying formulation conditions may include the preclinical candidate concentration, the excipients, or other formulation factors to examine their role in promoting conformational, colloidal, and thermal stability of the lead candidates. This row of septuplet sample conditions for the same candidate allows for a comparability assessment. The second scenario involves investigating deviations during manufacturing, whereby sampling the drug substance across the downstream bioprocessing workflow to identify risks through a multi-attribute comparability assessment. This information is critical, enabling a process control strategy to mitigate risk. This specific design of experiment slide cell array setup is ideal for an end-to-end comparability assessment of drug substance or drug product for risk mitigation, evaluating adverse impact on product quality due to process control changes, supply change, new manufacturing site, or simply a routine batch-to-batch comparability exercise in triplicate following the ICH Q5E global guideline for biologics. Our method involves the comparative analysis of weighted difference spectra for the reference standard against a drug substance or drug product. We typically determine the Euclidean distance at each wave number within the spectral region of interest to evaluate the two weighted spectral overlay differences and report the results in a table format along with the standard deviation. When identity is observed, then a highly similar or exact match occurs between the spectra within the full spectral region of 1800 to 1000 wave numbers. A test case like the one presented herein, where the reference is compared with itself, resulting in an identical match, denoting a Euclidean distance value of 0. Additionally, the 2T2D asynchronous plot does not have any contributing cross-peaks. Another possibility is when the absorbance difference is considered negligible, as defined by the target product quality attribute. The method can then be implemented to follow the ICH Q5E guideline as part of a comparability exercise. The implications may vary depending on the spectral region being evaluated. Let's consider the amide 1 and 2 band regions, where the amide 1 provides differences in secondary structure, solvent accessible surface area, and aggregate or self-association presence in one sample when compared to another within the same slide cell array. This multi-attribute comparability assessment is discerned in a comprehensive report. While the amide 2 band provides differences in amino acid composition, based on the 11 of 20 types of amino acids that can be detected, thus allowing for the comparability assessment of variants. The analysis of these two amide bands within the spectral region of 1,800 to 1,485 wave numbers provides an in-depth understanding of the relationship of multiple critical quality attributes based on the quality target product profile to ensure the product quality attributes needed. When evaluating the spectra within the glycan spectral region of 1,188 to 1,010 wave numbers, the carbohydrate-associated vibrational modes allow for the assessment of comparative differences between two protein samples to establish differences or similarities in the extent of glycosylation. The assignment of the cross peaks can be validated by the correlations established. Similarly, cross peaks associated with the excipients can also be used to confirm formulation conditions. The results can be interpreted using bar graphs, where positive values indicate regions of the first sample protein with greater content for the specific contribution, and negative values represent other regions where the second sample has a greater contribution. These bar graphs also inform which sample has a greater contribution when compared to another sample, or a reference standard in the same slide cell array. Contributions that may be associated with the secondary structure, amino acid composition, SASA, or glycan profile. Regions of the protein samples that are the same would have a near-zero difference and can also be observed, as highlighted. Ideal for developability and formulation optimization. The bar graphs summarize the quantitative nature of the analysis. The enhanced spectral resolution provided by the asynchronous plot allows for an in-depth evaluation. The negative bars indicate regions of the sample protein that have greater content for the specific crosspeak coordinate, such as the secondary structure, amino acid composition, SASA, or glycan. More importantly, the graph also provides regions of the protein that are the same, as highlighted. Ideal for ICH Q5E comparability exercise.